Okay, let's talk about the urinary system. Organs include 2 kidneys, 2 ureters, a bladder, and a urethra. So a lot less organs to discuss in this lecture than what we, especially in our very last lecture where we talked about the digestive system. Talked about a lot of organs there. Here we're going to, simplify things down to just 6 total organs. Now, the kidneys, fairly complicated little organs, and they are kind of the primary organs. I shouldn't even say they are the primary organs of our urinary system. They do most of the work. Really, everything else just either carries the urine or holds it. Our kidneys serve kind of 2 basic purposes: maintaining homeostasis by ridding our body of wastes, recycling necessary elements, so like things that we need to put back in the body, and then just other things that are not related to waste elimination and recycling processes, which includes maintaining blood volume. Volume in this case just means amount of blood, which, if you think back to our discussion on blood, we learned about half of it is water. So by controlling how much water we pee out or how much water we don't pee out, we adjust our blood volume. That's going to affect our blood pressure. Our kidneys also release a chemical called renin, which is an enzyme that activates a very complicated series of events, all of which increase our blood pressure. It helps maintain normal blood composition. Composition in this case means thinking back to the percentages, making sure that it's like 55% plasma, 45% formed elements. We have the correct amount of ions and stuff like that in there. Maintains normal body and blood pH. This is a very important way that we regulate the amount of acids and bases in our body. And it's really important for long-term pH balance. The other pH balancing system would be our respiratory system that we recently talked about. 2 hormones that our kidneys release include calcitriol, which helps us regulate our blood calcium levels, and then erythropoietin, or EPO for short, which is a hormone that helps regulate the production of our red blood cells. So pretty important organs, despite the fact that they're pretty small. We'll spend the majority of our lecture talking about them. Then we'll talk about, I think we just spent like 2 slides talking about the others, maybe 3 slides. Ureters, pretty much just a set of tubes, one from each kidney, that carries the urine down to the bladder. The bladder is just like a holding tank that holds the urine until it's time for disposal. And when it's convenient, it expels the urine out into the urethra, which is the structure. It's another tube that carries the urine to the outside of the body. Now, the urethra in a female, which is what's pictured down here at the bottom, is a singular structure in that it only functions to carry urine. The urethra in a man is a shared conduit. It carries not only urine to the urinary system, but it also helps carry semen through the penis during sexual acts. So we'll talk about the urethra of a man both in this lecture and then later on in the week when we move into the male reproductive system. This is not my favorite image, but it does show, the 6 organs. So I do want you to make sure you know it. What I want to have you focus on is just the stuff that's in capital letters. So the right kidney, the right ureter, the bladder, the urethra, left kidney, left ureter. Once again, make sure you keep your head straight on what's left and what's right. It's their right, their left. We've talked about other things before, like back when we talked about the aorta, we colored the renal arteries. Renal veins are also visible here in blue. We'll actually kind of talk a little bit about that here in just a little bit. Talked about the adrenal glands that sit on top of the kidneys. So I'm not going to worry about you knowing those. And then these things down here, the uterus, the ovaries, the fallopian tubes, we'll be getting into all that in the next lecture. So I'm not going to worry about those right now. Okay, let's talk more about the kidneys. Okay. Paired kidneys are described as reddish kidney bean-shaped organs located just above our waist, considered to be retroperitoneal, which we talked about in our last lecture. Means that they're behind or posterior to the peritoneum, in our abdominal or posterior to our abdominal cavity. They lie in that back abdominal wall. They're going to be on the right and left of our spinal column and are protected by our 11th and 12th ribs. But kind of more so the thick back muscles, especially those big lat dorsi muscles that we learned about previously, and that's why they're really susceptible. Just like you think about like boxers, they get they call it kidney punch, hit them in the lower back, can damage the kidney because there's just not as much bone protecting them. The 11th and 12th ribs are not going to protect the entirety of your kidneys. Right kidney slits just slightly lower. If I go back to this previous page, it's not noticeably smaller or lower, but it is a little bit lower, and that's because of thinking back to our last lecture, that right lobe of the liver, occupies a big space here in our upper abdominal cavity, and so the right kidney must sit lower because of that reason. Size-wise, kidneys are about 10 to 12 centimeters or 4 to 5 inches in length, 5 to 7 centimeters, 2 to 3 inches wide, 3 centimeters or about an inch thick. So not super, super big. They weigh about 4.5 to 5 ounces, which is pretty light. They're protected by 3 layers of tissue. So aside from those muscles and bones, 3 layers of tissue surround them. The renal capsule, now keep in mind we're going to work inside out here. So this is the innermost, deepest. This is membrane up dense irregular connective tissue, continuous with the outer coat of our ureters, and functions as a barrier against trauma, but also helps maintain the shape of our kidneys. Looking at our 2 figures here at the bottom, this is essentially just an artistic version of an actual dissected cadaver kidney, like we show over here on the right. You can see the very thin shell on the outside here where we've got the front removed from the kidney. That very thin outer protective layer is that renal capsule we show in both pictures. Okay. We don't show the other 2 in these 2 pictures. That's okay. The adipose capsule is in between the renal capsule and the renal fascia. It is a middle layer, so between those 2, it's made up of fat. Remember, adipose is just a fancy word for fat tissue. Protects our kidneys from trauma, holds them in place with the abdominal cavity. So it's kind of a fatty cushion. And then the outermost or superficial most layer is the renal fascia, a thin layer up into regular connective tissue. Helps anchor the kidneys and those adrenal glands on top to the surrounding structures on the wall. Now, deep to the renal capsule is the parenchyma of the kidneys. The word parenchyma is just a fancy word for the functioning part. In this case, it's glandular tissue that forms our urine. And the 2 divisions or 2 zones include a renal cortex and a renal medulla. I really like this picture because they color code it, showing kind of here in this light tan on the outside is the renal cortex. Thinking back to the very first lecture of the semester, we learned that cortex or cortical always refers to the outside of an organ. And then shown here in dark, the renal medulla, which is the inner part of the kidneys. And remember, medulla or medullary always refers to the inner part of an organ, in this case, the kidneys. Now, looking at our picture, we can see that the inner part of the kidney is not dark all the way through, that there are these kind of tan shaded extensions of the renal cortex projecting in between sections of the renal medulla. Each of those little column-shaped structures were literally called a renal column. Okay, so those project toward the center and divide that inner renal medulla into what we call renal pyramids. Okay, so each of these little triangular or fan-shaped areas is a renal pyramid. Okay, as you can see, if you follow the line, there's one renal pyramid. Let's see. A renal lobe consists of a renal pyramid, its overlying area of renal cortex, and then 1/2 of each adjacent renal column. So shown here in this boxed out area This is what a renal lobe is. One triangle shaped renal medulla pyramid, a half of a cortex on each, or half of a column on each side, and then the outside renal cortex. That's what one renal lobe is. You add them all up, there's about 6 to 10 of them total, because there's 6 to 10 renal pyramids. Within not only the cortex, but also the medulla, so throughout essentially the entirety of the interior of a kidney are these microscopic structures called nephrons. Now, if you look over here in our picture from the cadaver, you can't identify a nephron anywhere, and that's because these are microscopic in size. But these are the filtering structures which actually form the urine. Okay. And each kidney has about 1 million of these, a little over 1 million typically, where the urine is formed and then flows out of them in a specific pattern. And that's really nicely shown here in our picture from the cadaver. Basically, it's just a duct system. We have collecting the urine from the nephrons at what we call collecting ducts that drain into what we call papillary ducts, which come together and form what's called a minor calyx. Now, calyx is singular, calyces is plural, as it says. Okay. Each of these little cup-like structures is a minor calyx. They come together, several of them, and form a major calyx. We can see over here. Those major calyces then merge and form what's called a renal pelvis. Okay. The renal pelvis, like what's shown over here, is just the beginning of the tube known as the ureter that then carries that urine down, obviously, to our bladder. The renal pelvis extends out of the kidney in this indented area of the kidney, what we call its renal hilum. Okay, thinking back to our lecture on the lungs, and thinking back to when we talked about lymph nodes, and where a hilum in this case is just the indented portion of an organ. And this is also where renal arteries are able to carry blood into the kidney. Renal veins are able to carry the blood out. There's also nerves, lymphatic vessels, and so on and so forth. And a hilum expands into a cavity within the kidneys called a renal sinus. Okay? And the renal sinus is labeled here. Kind of difficult to explain. It's just a cavity where, it's kind of covered up by all that stuff there shown in yellow. But it basically would just be all this kind of area in the picture on the right. As far as the pictures go, don't worry about the cadaver version, but I would say everything else in this image would absolutely be fair game. And if we haven't talked about it yet, we definitely will. And so make sure you put a big fat star on this picture. It's a big important one. Okay, let's talk about the flow of blood to and from our kidneys. Something that's very, very important. Okay, although our kidneys account for only 0.4% of our overall total body weight, they receive about 21% of our blood pumped by the heart per minute, or 20% of our cardiac output. And now, keep in mind, this is a statistic that is only the case like when we're resting. So like when you're sitting around listening to me talk, if you're exercising, we drastically reduce the amount of blood that we send to our kidneys. Because at that point, it's more important to get that blood to our working skeletal muscle, our working heart muscle. But at rest, about almost a quarter of our blood goes to our very small organs known as kidneys. Thinking back to our lecture where we colored the aorta, we colored two small looking arteries down there in our abdominal pelvic region, what we call the renal arteries, which come directly off the aorta, come almost all the way into the kidneys, but just before they enter, they fork, okay? And each of them divides and forms what's called segmental arteries. Okay? The segmental arteries then give rise to what we'll call interlobar arteries, which are called interlobar because they travel between our lobes. Remember, inter means between, and remember a lobe would be, what I'm circling right now, one of those pyramids, a half a column on each side in the cortex. Interlobar arteries are going right in there through that area, through the renal columns, from the inside of the kidney towards the outside of the kidney. And zooming back out so I can keep reading. So the segmental arteries give rise to interlobar arteries traveling between each renal pyramid. Then they branch to form what we call arcuate arteries, and then what we call interlobular or cortical radiate arteries. And they're labeled down here as cortical radiate arteries, and here's kind of a flow chart to help keep your head straight. And those pass upwards and out towards the cortex, or into the cortex of the kidney. As that interlobular artery or cortical radiate artery ascends through the cortex, it branches into a series of what we call afferent arterioles, which it says as previously discussed. What I actually did was I moved this slide up, so it hasn't been discussed. But those then turn into a spherical mass of capillaries called glomerulus, or as they're labeled down here as glomerular capillaries, which are found inside of our nephrons. And so what we're doing here is we're zooming in this picture to show blood comes into, and this is a nephron. We'll talk about nephrons on the next page. Okay, glomerulus or glomerular capillaries are where we filter our blood and begin the steps in making urine. And we pull stuff out of the blood into this yellow tube-like structure. But we don't pull everything out. What's still going to remain in the blood leaves in what's called an efferent arteriole. Okay? That efferent arteriole leads to a plexus of peritubular capillaries, which are going to encircle, surround, in other words, that yellow squiggly line tube. Okay? We call those peritubular capillaries, and what they allow for is for us to pick up water and solutes that we're reabsorbing from the renal tubules. So essentially we put a lot of that stuff out or back in the blood that we pulled out. And then those capillaries then drain into our interlobular veins, and then we just go the opposite direction all the way out the kidneys by way of those renal veins. Now, surrounding what we call our loop of Henle, which we'll talk about on the next page, a segment of our nephron. It's a specialized set of blood vessels called vasa recta, which are not shown here in our kind of flow chart, but they encircle the loop of Henle, crossing between what we call the ascending and descending limbs. And all this, really small stuff will make more sense here after we get through our next couple slides. Renal veins, like I said, basically travel the same path in reverse as the arteries. So from the interlobular cortical radiate veins to the arcuate to the interlobar to the segmental, they successfully merge and exit as a single renal vein, usually one from each kidney, drains and then into that inferior vena cava that we've previously learned about. As far as the image here on the left goes, it's way too hard to identify and label things. But I definitely think it's a good idea for you to kind of know this. It's kind of a pun here, but flow chart, because it's important to kind of understand the order in which the blood is flowing. Here next to cortical radiate arteries and cortical radiate veins, might not be a bad idea. Put their other name, interlobular artery, and then interlobular vein. And veins, because there's not just one. So, interlobular was the term that the previous textbook edition kind of went with. They kind of changed it. I guess it makes sense. Interlobular and interlobar are so similar sounding that it's probably confusing. Make sure you know them both ways, though. Over on the right is an illustration. Actually, it's not an illustration. It's a picture of an actual kidney where they've removed all that glandular tissue, so all the the functioning part of the kidneys and all that's left, and they've injected it with a really cool substance that hardened up all those blood vessels, which just demonstrates how dang crazy the vascularization is within a kidney. Essentially, every square centimeter is receiving blood flow, and really, really neat. But not something I'm going to ask you to know. I just think it's pretty cool to see. Okay, let's talk about those nephrons. All right, kidneys contain about 1.2 million functional units called nephrons. The units that form our urine, and each of those nephrons consists of two principal parts: the renal corpuscle and a renal tubule. The renal corpuscle I talked a little bit about on the previous page, is where we filter our blood, pulling out a lot of the fluid from our blood plasma, along with some of those very important dissolved substances like sodium and potassium ions and glucose and other things. Each renal corpuscle consists of an afferent arteriole, a glomerulus, and an efferent arteriole. The kind of three things I was talking about back here on this previous page. Okay, looking at our figure here on the bottom left is a fairly simple illustration of a nephron. And I say fairly simple because if I go to the next page, we show there's kind of a lot more that go into it. And here's a zoomed in version that we'll reference here quite a bit in just a minute. The other part of nephron is this long winding, what we call renal tubule, which is going to finalize and process that filter plasma and form our urine. And we'll get into the renal tubule anatomy here on the next page. For now, let's talk about the structure of the renal corpuscle. As we said, there are three major blood vessels associated with each renal corpuscle: the afferent arteriole, the glomerular capillaries or glomerulus, and then the efferent arteriole. Okay. That afferent arteriole, we learned, is a distant branch from our renal arteries that carries the blood into this structure, the entire structure being our renal corpuscle, okay, which we've shown internally but is enclosed, like you show here. It's basically a bubble with capillaries on the inside. Okay. That mass of capillaries is called a glomerulus and is able to function the way it does due to a large pressure gradient. The pressure exists inside of those capillaries that is going to help push that water and the small substances out and into this capsular space, or as it's sometimes described as a Bowman's space, which at this point, it's not blood because we aren't removing the blood cells and the bigger stuff from the blood. It's basically plasma with water and solutes and other stuff that we will call filtrate or glomerular filtrate. Okay. So blood in the glomerular capillaries is under high pressure due to having branched from the aorta. So substances in our blood that are small enough to pass through the pores in the capillary walls are forced out into the Bowman's capsule. Okay. Looking at a very small segment of this picture in the middle there, we show that the capillary wall of those glomerular capillaries are fenestrated. Now, we just talked about blood vessels here about 2 weeks ago, or maybe a week and a half ago. We learned that fenestrated capillaries are really, really permeable to substances like water because they've got these big holes in them. Think of them like a colander that you dump your macaroni and your water into when you're getting ready to make mac and cheese. The water is going to be able to pass directly through those holes very easily, whereas the big noodles, those are not going to be able to pass through those tiny holes. Now, to prevent other stuff from going out of the blood that really shouldn't, directly surrounding the endothelial layer of the glomerular capillaries is that basement membrane that all capillaries have. But there's also what we call pedicels, or it's going to be those podocytes, which have pedicels. Each of these little yellow, they kind of look like bugs to me, is a cell called a podocyte. Okay. Podocytes wrap around those capillaries and have these little fine projections called pedicels that mesh with the adjacent podocytes. So they basically, it's kind of like locking fingers together, wrapping around the glomerular capillaries. And there's a little membrane that goes across the little space in there, which is called a slit membrane. And it allows, as it says, medium-sized proteins from passing through. It's kind of, to me, like having a coffee filter that surrounds, another coffee filter that surrounds the capillary wall. So it's going to allow water and very small substances out into that space, but not allow everything out into that space. What we don't want is red blood cells, white blood cells, platelets, and proteins getting lost in our urine. You should never have blood in your urine. That means something's wrong with you. Okay? Now, those cells called podocytes are one of the two types of cells specific to our glomerular Bowman's capsule. A two-layered capsule. One of those is the podocytes. The other is a layer of simple squamous epithelium, which is what we have here is like the parietal layer. Basically just encloses the entire structure. Okay? So there's the parietal layer, then there's the visceral layer, which is what the podocytes are. Okay? Capsular space is the space where that filtrate collects before entering into the first segment of our renal tubule, what we call the proximal tubule or proximal convoluted tubule, as it's labeled there. What's left in the blood leaves the capsule or in our efferent arterioles. Okay. Now, I do want you to know this figure. It's not my favorite image, but there are some very important things that are labeled here. So highlight the following: the afferent arteriole, the parietal layer of the glomerular capsule, the capsular space. Just highlight podocyte of the visceral layer. It's too kind of difficult to label the pedicel. The pedicel is a little leg-like structures on the podocyte. It was kind of hard to tell them apart here in this picture. I'm not going to worry about this picture, and I'm going to worry about this picture. So just make sure you know those parts. Okay. Now, this is just a zoom in at this little segment of our nephron. We still have quite a bit to talk about for nephron because we haven't covered, but we are going to right now talk about the renal tubule. All right, let's talk about the renal tubule. A winding hollow tube extends from the corpuscle to the end of the nephron, where it joins a collecting duct, which is shared in common with other nearby nephrons. It is described as a urine collecting and concentrating tube, and is divided into three regions: the proximal convoluted tubule, or sometimes you'll just see this listed as a proximal tubule, a loop of Henle, which is also known as a nephron loop, and then a distal convoluted tubule. Sometimes you'll just see it listed as a distal tubule. I'm going to go ahead and talk about collecting ducts here on this page because they are closely tied in with nephrons, as I've already kind of talked about. Looking at the bottom of the page here, we show that there are two different types of nephrons that exist. On the left here is a cortical nephron, which is the majority of the nephrons that we have in our body. Was it 85% of the nephrons in our kidneys? Difference between them and these, what we call juxtamedullary nephrons, is that a cortical nephron is pretty much, the majority of it anyway, is out in the renal cortex. Cortical for cortex. Juxtamedullary has a really deep down dip because of the loop of Henle. So it's pretty much a big component of our renal medulla. Okay. They only represent about 15% of the nephrons in the body. Okay, so let's talk about the first segment of our renal tubule, what we call the proximal convoluted or proximal tubule. The word convoluted that I had up there above is a fancy word for like jumbled up, which is exactly what this tube section is. A kind of jumbled up, twisting, turning tubule that receives that filtrate that was formed back in our corpuscle. And within the proximal convoluted tubule, we have a layer of cuboidal shaped cells, a single layer, simple cuboidal, with a lot of mitochondria inside. And the cells have a prominent microvillus, which we could actually see here on the previous page. Remember, microvilli are the little finger-like extensions. We call this layer of microvilli within our kidneys the brush border, which is just like the brush border that was found in our small intestines that we recently talked about. And the fact that it's got a large access to energy from the mitochondria and a brush border make it really good at performing what's called reabsorption as well as secretion. And it's kind of crazy, but what we do back here on this previous page, we talked about forming filtrate. We're going to pull out like gallons and gallons and gallons of water from our blood within that corpuscle every day. Now, we don't pee out nearly that much amount of urine, which means most of that water and a lot of that stuff that goes with the water actually gets put back into our bloodstream. What our proximal convoluted tubule does is actually is where most of that water returns back to the blood. So it seems kind of stupid. We pull out a lot of the water and stuff here, but then we give it back right there. It's stupid, but that's what we do. I don't really understand it, but that's what the situation is. And reabsorption is where that water returns from the tubule back into the blood. Secretion will be the opposite, where we do push stuff out of the blood into the tubule. The location of the proximal convoluted tubule is strictly out in the renal cortex. Our next segment is called the loop of Henle or nephron loop, a long U-shaped structure. The cells of our loop of Henle can vary from simple squamous, so a single layer of flat plate-like cells. Some are simple cuboidal, or some areas have simple cuboidal, single layer of cube-shaped cells, and some areas have simple columnar, so a single layer of column-shaped cells. Here, the cells almost entirely lack microvilli, so they're not like the cells in the proximal convoluted tubule. Looking at our figure, if I kind of zoom in, notice down here there are two segments to our loop of Henle, a descending limb and an ascending limb. The descending limb is going to go down into the medulla. The ascending limb turns around and goes right back out into the cortex. Okay? Now, looking at the descending limb versus the ascending limb, it's very subtle. Over here, we can kind of see it a lot better. The descending limb is a lot thinner than the ascending limb, which is a lot wider. We call these thick and thin segments. Okay? And this is important because it relates to the movement of water. Okay, so let me read through. Thick segments of the loop have simple cuboid epithelium and are engaged in active transport of salts, and therefore need extra mitochondria for ATP production, making the cells more thick. The thin segments of the loop have simple squamous epithelium and are very permeable water. So the water is able to easily pass through them. There are two limbs to each loop. The descending limb passes from the cortex and the medulla, contains a thin segment that suddenly narrows as it descends. The ascending limb is a portion of the loop turns 180 degrees and returns to the cortex, and it contains that thick segment that widens the tube as it passes upward. So essentially, In the descending limb, what we're doing is allowing water to escape the tubule, and it's going to jump right back into all those blood vessels that are surrounding it. The ascending limb, it's not going to be moving water, but instead is pumping out salt into that renal medulla. This is why they say if you were to ever eat or taste a kidney, the really centered part of it will be really salty. Okay. Now looking over here on the right, that very long loop of Henle is surrounded by a blood vessel that I talked about on the previous page, what we call the vasa recta. And they don't give it a special name over here. It's just what we call peritubular capillaries. From our ascending limb of the loop of Henle, the filtrate is going to flow into what we call the distal convoluted tubule, which is just distal to the loop of Henle and functions to conduct the filtrate out of the nephron into a collecting duct. Location of a distal convoluted tubule is strictly out in the renal cortex. Now, zooming in, right here we see a segment of that distal tubule passing right by that afferent arteriole. On the next page, where I'm going to have you highlight some stuff, go ahead and draw a circle right here like I'm doing with my cursor. Right here where that distal convoluted tubule and the afferent arteriole are passing by one another, and draw a line and label that as what we call the juxtaglomerular apparatus. And that is a portion of the distal convoluted tubule that can communicate with that afferent arteriole. With that communication, that helps maintain the homeostasis of blood flow into our nephron. Now, if I go back to this page right here, I didn't talk about this yellow oval shaped area in our picture, but notice it's labeled here as the ascending lobe of Henle. I don't know why, because it's really more so the distal convoluted tubule. But this is the tubule, and it's actually located right up next to and touching that afferent arteriole. These cells here of the afferent arteriole, called juxtaglomerular cells, and these cells of the tubule, called macula densa, actually communicate with one another. And what this allows for is basically to, if we need to, if we're like moving stuff way too quickly and we're going to end up like peeing too much stuff out, we can send a signal from the tubule to that afferent arteriole to decrease the amount of blood that flows into the nephron, and that slows down the formation of that filtrate, so we can get the stuff back into our blood that we need to, and we won't lose it in our urine. Okay? So that's what the juxtaglomerular apparatus does. I'm not going to really read through all that because it's a lot of physiology, but I just kind of wanted to give you a breakdown of what's taking place. From our distal convoluted tubule, the filtrate is going to flow into what we call a collecting duct, which is formed by joining the distal convoluted tubules of several nephrons. So a collecting duct actually collects filtrate from multiple nephrons. You can kind of see these little branches here, kind of going nowhere, which is actually, the picture is just cut off. There'll be another nephron connected over there, another one over there, another one over there. So collecting ducts collecting the filtrate from multiple nephrons. Okay. Within a collecting duct are cuboidal shaped cells with very few microvilli. They, within the medullary pyramids, merge and form what we call papillary ducts. At this point, papillary ducts, actually shown down here on the next page, we can stop calling it filtrate. We can still start calling it urine. That urine is then going to flow down and drain into those minor calyces that we talked about here just a couple of slides ago and located in our medullary papilla. Two types of cells are present in the collecting duct, but also the final portions of our distal convoluted tubule. Principal cells that respond to the two hormones, aldosterone and antidiuretic hormone, and the intercalated cells that secrete hydrogen ions to help control our pH. Okay, I'm not going to worry about identifying these two pictures here, because this one I included a full-size version where we can really see everything. This is a cortical nephron, which once again is the majority of the nephrons in our body. So highlight in this picture the following. The proximal convoluted tubule here on the left, all those little red squiggly lines, which are the paratubular capillaries. Over on the right side of the image, highlight the corpuscle itself, but also the capsule and the glomerulus is part of it. The efferent arteriole, which you can tell is a lot smaller than that efferent arteriole, so that's an easy way to tell them apart. So efferent arteriole, the distal convoluted tubule, the afferent arteriole. Make sure you did circle and draw in the juxtaglomerular apparatus. Then you can skip down to the nephron loop, which it might be a good idea in parentheses, put in parentheses loop of Henle, because that's what it's also known as. That's kind of what I usually call it. And then highlight descending limb, ascending limb, collecting duct, papillary duct, and renal papilla. Might not also be a bad idea to kind of draw a big old star around this box. Keep your head straight on the flow of fluid through a cortical nephron, because that's important. Okay. We will stop here, which is perfect spot to stop. We've got 2 slides left, which will be easy to finish up on Thursday or Wednesday, excuse me. We'll pick up with ureters, which collect that urine from our kidneys, and then we'll get into our bladder and urethra. So we will stop here.